Master 6 BSc Botany Honours student. Here I am Dr.A.S.Khalkho. I deliver a lecture on photorespiration. It is also known as C2 cycle. Earlier it was believed that the rate of respiration in light was almost equal to the respiration in darkness. Recently it has been observed that light affects respiration and the rate of respiration in light is about 3 to 5 times more than the respiration in darkness. That lead to the discovery photorespiration. It is also known as C2 cycle. The existence of photorespiration was first demonstrated by Decker in the year 1955 and 1959. He and his associates were the first to use the term photorespiration. Definition: the respiration that occurs in green cells in presence of light resulting into excess evolution of carbon dioxide, or it may be simply defined as the release of carbon dioxide in respiration in presence of light. The photorespiration has been reported in green cells of different plants like Nicotiana, Pisum, capsicum, and in algae Chlorella and Nigella. Photorespiration always requires light and its rate is maximum between 25 degree and 35 degree centigrade. It also depends on the oxygen concentration. Site of photorespiration. According to Hui and Crotier, 1968, site of photorespiration is chloroplast. And according to Tolbert et al. 1968, discovery of perisome containing enzymes of glycolate metabolism suggested a correlation between the perisome and photorespiration. Third, Tolbert 1971 proposed that carbon dioxide evolution during photorespiration takes place in mitochondria. The above findings of various workers indicate a close relationship among chloroplasts, perisomes, and mitochondria. At present, most of the physiologists believe that chloroplast, perisome, and mitochondria, all the three cells organelles, participate in photorespiration, and this is the diagrammatic representation of various steps of photorespiration. This is the mitochondria, chloroplast, and perisome. Biochemistry of photorespiration. In photorespiration, chloroplast, mitochondria, and perisomes are involved. They are found in close association with each other. Third, in chloroplast, when photosynthesis occurs, an early product, glycolate, is produced, which is used as primary substrate for photorespiration. Glycolic acid is produced as a result of oxidation of ribulose diphosphate when the concentration of carbon dioxide in the external atmosphere is less than 1%. Fourth, at first, ribulose diphosphate is oxidized into 3-phosphoglycic acid and 2-phosphoglycic acid in presence of enzyme ribulose diphosphate carboxylase, also known as ribulose oxygenase. This is the equation: ribulose diphosphate, 3-phosphoglycic product is 3-phosphoglycic acid and 2-phosphoglycic acid. Fifth, 2-phosphoglycic acid lose phosphate group to form glycolic acid. 6. The glycolic acid, thus synthesized in the chloroplast, is transferred to the perisome where it is oxidized into glycosylic acid and hydrogen peroxide in the presence of enzyme glycolic acid oxidase. This reaction was worked out by Geltics 1966. This is the equation of this reaction. 7. The hydrogen peroxide is catalyzed into water and oxygen in presence of enzyme catalase. 8. The glycolic acid formed as a result of oxidation of glycolic acid is converted into an amino acid glycine in presence of enzyme glucotic glycosamintransferase is the equation. Glycosylate is the enzyme and form the glycine. Ninth, the glycine amino acid moves into the mitochondria where two molecules of glycine interact to form a molecules of serine amino acid and one molecules of carbon dioxide. The carbon dioxide is then released in photorespiration. Two molecules of glycine and one molecules of serine and form one molecules of carbon dioxide. The serine is then transported out from mitochondria to the cytoplasm where it is converted into hydroxypyruvate and then into phosphoglycerate which enters into Calvin cycle and form glucose or carbohydrate. And this is the diagram of the whole reaction: mitochondria, chloroplast, and very soon is the completed photorespiration. Thank you.